So Bio 30, we're moving on to chapter 12 now on the nervous system, sensory receptors and sensory perception. I'm going to break this lesson into three different segments. The first one is going to be on chemical sensation and sensations involving your skin, and the other two will be more specifically on vision, and the last one on hearing and on balance. So first of all, when we do talk about sensory receptors, what we are dealing with is measuring things either from outside of your body or inside of your body. You need to be able to detect various different stimuli before you can actually send it to the central nervous system. So of the different stimuli that can be detected from inside or outside of your body, keep in mind that your nervous system really only understands one kind of message, and that is the electrochemical message. So we do have to have some sort of energy transduction taking place, be it from mechanical energy to electrochemical energy, or from light energy to electrochemical messages. It is the brain that the message ultimately does reach, and the region of the brain where that message reaches really determines the perception that is detected. So if you are taking in chemical information in the form of smell, that goes to a region of your brain that we'll learn a little bit more about in the days to come, but a region of your brain that's referred to as the temporal lobe. If we're talking about light information transmitted from the eyes toward the brain, it goes to the very back of the brain, to the occipital lobe, and that is where that information is processed. Now, everyone's brain is a little bit different. It's different due to genetics, it's due to differences in embryonic development as the brain is growing and maturing, and it also has to do with development of the brain from the point that you were born as well. So because everyone's brain is a little bit different, we do in fact see things a little bit differently. So as it says in this quote here, we do not see things as they are, we see things as we are. Even though we may all be looking at, hearing, seeing, smelling exactly the same thing, we don't necessarily perceive it in exactly the same way because each of our brains, again, are going to be unique. So how do we actually take information from one form and convert it into the form that your nervous system can understand? Well, what you do have to have are various different kinds of sensory receptors, and they do exist in several different forms. If we take a look at the bottom here, what we have are three different sensory neurons. We can tell, of course, that they're sensory neurons because they have the more centrally located cell body. The first one that we have on the left-hand side, this is the simplest. This one here, it just says that we have free endings of the sensory neuron. That is where the action potential is going to be generated in response to a specific kind of stimuli. And then, as with all neurons, it will pass down from one end to the other, to the axon terminal end. The one in the middle looks a little bit different. Here at the dendrite end, at the receiving end, it's a little bit more specialized. So this looks like one that we would find in the skin and perhaps is for touch or for pressure. So when there is touch or pressure in the skin, it's going to activate this specialized ending of the sensory neuron, generate an action potential. And again, in the end, the result is the same. It sends an action potential down along the length of the neuron. The last one that we have here, different again. In this case, there is a completely different receptor cells. So we have two different cells involved. We do have the initial receptor cell that will generate an action potential, but then that action potential does have to cross a synapse to pass it on to the sensory neuron. So the sensory neuron in this last example is completely different than the sensory neuron. And an example of where we would see that is in the eyes and also in the ears, dealing with hearing. Several different kinds of sensory receptors then, and they can detect various different stimuli. Again, these stimuli can be coming from outside of the body, but you also have to detect things inside of your body. So based upon the energy transduced, sensory receptors, they fall into five different categories. Now keep in mind, as I mentioned, that for all of these different incoming energy sources, all of them do have to be converted into an electrochemical message and action potential that is eventually sent to the central nervous system. Your central nervous system itself does not respond to heat or cold or light or pressure or anything else. It only responds to an action potential that reaches that region of the brain. But scattered throughout your body, you do have these various different kinds of receptors, or at least most of these receptors. Mechanoreceptors you have, for example, in your skin. They respond to things like touch and pressure and pain, and also in and around your ears for the sensations of hearing and balance. Chemoreceptors, a couple that you have, taking information from outside of your body for the chemical sensations of smell and taste, but also inside of your body you do have specialized receptors for measuring gases in your body like oxygen, carbon dioxide, and also hydrogen ions. Photoreceptors, very, very specialized. They detect only one thing, and that is light. Electromagnetic receptors for measuring magnetic fields. We know for sure that other animals do have these for measuring magnetic fields, especially migratory animals like birds and whales, and possibly humans have these as well. Thermoreceptors, again, you have these in your skin, specialized receptors for detecting heat and completely separate ones for cold. And you do have pain receptors as well. The technical name for them, nociceptors, and they respond to excessive heat, excessive pressure, and to chemicals. So if we do have an increased intensity of the stimuli, as we know, that cannot increase the size of the action potentials. Remember, action potentials, they are all or none. So if we have a stronger stimuli, the size of the action potential remains exactly the same. So if the size of the action potential, let's just draw a graph here again to remind us all what that looks like. So we're plotting time versus voltage. This is our resting membrane potential, and of course we have the threshold potential. So if there is enough depolarization to reach the threshold potential, there is an action potential. What happens if there is a stronger stimuli? Well, you reach the threshold, you get an action potential, but again, it's exactly the same, at least those lines should be at the same level, the same strength, the all or none 100% action potential. So how you distinguish then between different intensities, how you tell if something has an increased intensity, a couple of different possibilities. You can increase the frequency of the action potentials. So we can bring those peaks much, much closer together. We can get one action potential right after the other, and that would be in response to increased intensity. Another possibility is increasing the number of receptors that are stimulated and the number of neurons that are sending a message to the brain. So due to these two different processes, this is how we can get what is called a graded response and be able to distinguish different intensities of the stimuli. We may also find some neurons have a different threshold level. So using the same diagram here, there may be other neurons that have a threshold higher up. That means more depolarization is required before an action potential is generated, and you would require a greater stimuli, a stronger stimuli or more intense stimuli in order to generate an action potential for those particular neurons. This picture here, we are taking a look not at a mammal or not even at a vertebrate, but we're taking a look at a crustacean here. And it is showing us that we do have a sensory neuron going from the left-hand side where we do have the dendrites receiving some information. And in this case, it's receiving information from the muscle, and it's going to be some stretching in the muscle. And we can see that the message then is then traveling along the length of the axon and eventually reaching the central nervous system of this crustacean. So if we take a look at the top, it says that what we have here is a weak stimulus, a slight bending. So if we do have a slight bending, if we take a look at the action potentials, take a look in particular at the frequency. of the action potentials and the spacing between each one of the action potentials. At the bottom, the only difference here is now we have a stronger stimulus. So how does this change the action potentials? Well, it doesn't change the height of them, right? They're exactly the same. They are all or none. But what we do have here is, of course, as we can see, a much higher frequency. Now we have brought together the peaks of those action potentials as close as we possibly can, minimize the refractory period. So this is in response to a stronger stimuli. So once again, action potentials the same size, higher frequency of the action potentials. We could also have many, many more neurons that are sending the information to the brain from a larger stimuli. And as I mentioned as well, some other neurons may not respond unless there is a larger stimuli and they would have a higher threshold. And yeah, so this is just a graph that is showing us that in fact we do have neurons that are at a different threshold level. So this one here, a lower threshold level would generate an action potential. For other neurons to generate an action potential, it shows a higher threshold for those ones. A little bit more terminology. The term that goes along with this one is adaptation. The idea behind this picture is you stare without blinking, without gazing from side to side or up or down, right at the very, very center where you see this darker black dot. Now initially you'll notice that there is a cloud around the outside that gets fainter and fainter the further away you go to the periphery or away from this black dot. What you should notice is if you do stare directly at the center, then what is in the periphery, all of that gray, it will gradually disappear. What is happening is the photoreceptors in your eyes, they're becoming accustomed to that stimuli and they're no longer responding to it. So continuous stimuli over a prolonged period of time, your receptors become accustomed to that, no longer respond to it, no longer generate an action potential, no longer send a message to the central nervous system, and this is then referred to as adaptation. The receptors have adapted to that constant stimuli and are no longer responding to it in the form of an action potential. So the two senses that we'll now go on and talk about are both chemical senses. The first one involves your nose, involving the sense of smell, also called olfaction. Humans are pretty impressive in a number of different ways. We can distinguish thousands of different smells. We do not have any separate receptor cells. So it's not a separate cell like we'll see with vision and like we'll see with hearing. Instead, what we have are specialized nerve endings with cilia on them. Now, these cilia are going to respond to chemicals that are moving in through the nose, but these chemicals do have to, first of all, dissolve in a liquid, and that liquid that they dissolve in is the mucus layer on the inside of your nose. We don't know as much about the sensation of smell as some of the other senses, but we think that there are in excess of 1,000 different receptors responding to different chemicals to different degrees. They have found that there's no particular pattern in your nose for where these different categories of receptors are located. So there aren't, for example, receptors for pungent odors toward the back of the nose and sweet odors toward the front of the nose. There is no particular pattern for them. Now, this number might seem kind of small, 3% of our genetic information devoted to receptors specifically for smell. In fact, this is quite a large number. And if we were to take a look at some other mammals, in fact, just about any other mammals, even if you think of some pets like dogs and cats, they have much, much keener smell than we do, and a much greater percentage, perhaps 5 to 10% of their genetic instructions would be dedicated to their sense of smell, and a much larger part of their brain as well would be dedicated to the sense of smell. Just an indication of how important that sensation is for them. And we probably lost a lot of that in genetic information as we become more dependent upon our sense of vision. And the last point that I have here, an individual odor molecule stimulates several types of receptors, probably to different degrees. And what it produces is, well, what's usually described as a code-like pattern for a particular fragrance or a particular scent. And that information then, of course, in the form of action potentials, is what is sent to the brain, and that's what is processed. In this case, in the temporal lobe of the brain, where that information is processed and interpreted as different smells or different fragrances. So a couple of pictures here showing the nasal cavity and the nose. So this is showing what we do have, some different receptors detecting different chemicals. So again, thousands, perhaps thousands of different kinds of receptors. So here, very, very simplified, they're showing this one receptor here that's kind of a square shape, this other receptor here, more of a circular shape, and it shows that it's different chemicals that are going to attach to each of these different receptors. Some chemicals will partially attach to some receptors, generating a weak action potential, or rather a weak or lower frequency of action potential. Other ones will bind more strongly. So again, depending upon what the chemicals attach to, what receptors are stimulated, where the information goes, the pattern that's generated in the brain, that determines the actual smell and the perception that the person has. Once again, these chemicals, they do need to dissolve in the liquid portion in your nose, which is the mucus portion of your nose. What it's also showing here, right up above the nasal cavity, you have a region of the brain that's sort of tucked underneath the front portion of your brain. It's called the olfactory bulb. It has it written here, the olfactory bulb. That is the first part, I guess, of the brain where the information goes, but quickly from there, it's directed toward the back, and it doesn't go directly to the frontal lobe, but it does go to the temporal lobe initially, and from there, it's directed to various other brain regions as well. Just a couple of other pictures here showing the underside of the brain. When you do the brain dissection, you can take a look and you can actually see these little flaps of tissue that are underneath the frontal lobe. So this is the underside of the brain. This is the front of the brain at the top. This is the back of the brain that we're taking a look at the bottom here. And that's the olfactory bulb. The temporal lobe in this case sort of tucked underneath as well. So this would be one of the temporal lobes here, it looks like. The other one over here that I'm kind of outlining. And the other image that we're taking a look at the right, again, just showing in this case in the yellow color where we can see the olfactory bulb. And this is going to be the temporal lobe region back here where that information is traveling to. So here it does say a few other brain regions. These ones, in fact, you don't need to know about, but you may come across them in your reading. So it does go to the limbic system, which is in fact part of the temporal lobe, other regions in the amygdala and the hippocampus. These are related to things like behavior, emotions, memory, and that's why certain smells and aromas can sometimes trigger different emotional responses and trigger some fairly strong memories as well. Now, there is another organ that is located in the nose. It's located in the nose of other mammals, and we think that maybe it's located in humans as well, although it hasn't been definitively recognized and identified in humans. This organ here is called the vomeronasal organ, and what it responds to are chemicals, but they're not chemicals that do have a specific scent or aroma or fragrance. In fact, they don't have a scent associated with them. These chemicals are given the name pheromones. And when these travel into the nose and bind to receptors in the vomeronasal organ, these are going to regulate hormones that are related to mating and social functions. And again, not entirely sure if that is the case in humans, but certainly it is the case with other mammals. The other chemical sensation that you do need to know a little bit about, and again, there's not a lot with these chemical sensations, a lot of detail that you do need to know. But the other one is your tongue, your mouth, the sensation of taste, also referred to as gustation. So in humans, not quite as complicated in a number of different ways probably than the sensation of smell. In the mouth and on the tongue, there are what are referred to as taste buds, and these are usually described as onion-shaped structures made up of about 50 to 100 cells. So if we take a look at the surface of the tongue here, there are all of these kind of pits going down into the tongue, and it's in those pits where they are lined with these taste buds. So this here is kind of the onion-shaped structure. So we would have to have, again, the chemical that's dissolving in a fluid, and the fluid in the mouth, of course, is saliva. So from there, we can have the chemicals dissolved in the fluid that's going to travel in to the taste bud area where it's going to bind to receptors, stimulate those receptors that are part of sensory neurons, generate an action potential, and again, send that information toward the brain. So let's just take a look at a little bit of a clip here. That's just going to show us some images of the tongue and of the taste buds. So surface of the tongue that we see here, and they're just going to zoom in. So now we can see those little pits or those little depressions. All along the surface, there will be saliva. And there's pointing out the taste buds. So we'll zoom in and take a look at those taste buds. So what it's showing at the top here are the chemicals traveling into your mouth, dissolving in the saliva that can now travel into where the taste buds. And they're activating or attaching to these sensory receptors or the specialized ends of these sensory neurons. Again, it says that it's dissolving in the saliva there. And there's the neurons and the nerve that's sending the information toward the brain. Well, this is one of the reasons why it's not quite as complicated as in the nose. In the nose, like I said, there are in excess of 1,000 different kinds of receptors. In the case of taste, there are only 5 of them. I'm sure you've heard of the first 4, sweet, sour, salt, and bitter. There is now recognized to be a 5th one. It is called umami, and it's usually described as a savory taste, a meaty taste. It's stimulated by the amino acid in proteins glutamate and MSG, monosodium glutamate, that will also stimulate that particular receptor umami. If you're the kind of person that likes hot and spicy food, it actually doesn't stimulate any of these sensory receptors. What it stimulates instead are the pain receptors. You do have pain receptors in your mouth as well, and if you do like that hot and spicy food, like the capsaicin from chili peppers, then it is activating those pain receptors in your mouth. Each one of the different taste cells, individual taste cells, does only have one single of those 5 different receptor types. But in a single taste bud, there are many different cells, and there can be various different combinations and proportions of these 5 different receptors. The picture that it's showing here is showing a map of the tongue, and this map of the tongue does in fact have a cross through it or a line drawn through it, because it's now recognized that unfortunately this picture that appeared in textbooks for probably 100 years is wrong. So what this picture does show is that you have bitter receptors at the back of your tongue, sweet right at the tip, and sour and salty toward the sides. Now, probably not entirely incorrect, but definitely not entirely correct either. What you do have are all of the 5 different receptors all over the tongue. Yes, there may be a greater proportion in one region of the tongue compared to the other, but it certainly is not the case that you only have bitter receptors toward the back and sweet receptors toward the front of the tongue. Just like in the nose, the chemical must first dissolve in a liquid, and that liquid is saliva. Hopefully you recognize that your sense of smell is very closely linked to your sense of smell, your sense of olfaction. So if you are stuffed up, if you do have a cold, then that does affect not only your smell, but it affects your taste as well. Also kind of interesting, if you don't like certain foods, it may in fact be because of your genetic makeup. They do now realize that what we do like in terms of our taste is in fact inherited from our parents. The next one that I'll talk about here, not chemical, but this is somatic sensation. So these are sensations that do typically involve your skin, possibly involving your muscles, your joints, and your tendons as well. So receptors that we have in our skin include for touch, pressure, heat, cold, and pain. And if we do take a look at a picture of the skin here, your skin, I'm not sure if you realize this, but it is the largest organ in your body, and it's also a very, very complex organ. It has not only nerves going all through it, neurons going through it, it has blood vessels going through it, various different glands that you have in the skin for oil and for sweat. But of course, you do have receptors, receptors that detect information and pass that information on to your brain. So as we can see here, you do have separate receptors for heat and cold. Those are separate sensory receptors that when stimulated by the appropriate stimuli generate an action potential and send a message to the brain. You also have receptors for light touch, gentle touch, and for more pressure, stronger pressure. And you have other ones which are specifically for pain. So once again, very, very complex, the structure of the skin and all of the different structures and processes going on with the skin. So you might think, well, what are the advantages of pain? Wouldn't it be better if you didn't have any pain receptors? Well, unfortunately, then you wouldn't know if there's some stimuli that could possibly damage your body. So if you think back to the reflex lab that we did, we said that the purpose of reflexes is to protect your body. So if we're testing this reflex here, what this rubber mallet is doing is it is stretching the tendon. It's stretching the patellar tendon, it's stretching the quadricep muscle, and if those structures are stretched too much, they could be damaged. So the response is to have a reflex action in order to protect your body. Well, it's kind of the same thing with pain. The pain is to protect your body as well. So hopefully, if there is some painful stimuli, then in this case, it will be conscious, unlike a reflex. But if there is some painful stimuli, then hopefully you will take your body away from that painful stimuli and learn in the future to avoid that painful stimuli. So like I said, we'll divide this into three different lessons. So we'll leave photoreceptors for the next lesson.